Good morning. Uh, it's really my pleasure this morning to introduce our speaker. Millions of patients, thousands of our patients, millions of patients worldwide have implantable pacemakers and defibrillators, and 99% of them rely on leads to detect arrhythmias and to deliver therapy. If the lead doesn't work, it's big trouble. And a major portion of the lead that has been a problem over the last 50 years is the insulation. Kim Chaffin is vice president at Medtronic. She's been there since 1999. She obtained her PhD in chemical engineering and material sciences from the University of Minnesota, where she had the great good fortune of being advised by Dr. Frank Bates, who's a Regent Professor of Chemical Engineering and Material Sciences, internationally famous. I just looked up his citations. He's been cited over 20,000 times in the literature. So Kim joined Medtronic, and there she has become a senior member of the team, and she's responsible for research projects into advanced technologies and material thermodynamics. She's married. She has two children. Her son is now a engineer at Boston Scientific, and her daughter is a chemical engineering student at the University of Michigan, and she is going to be taking her MCATs. So, Kim, thank you for coming this morning.
Dr. Hauser. John, my audio is on. Do you feel like for the online? Okay, good. Fantastic. Well, that was a nice introduction. Yes, I just came from Parents Weekend at the University of Michigan, and my crazy daughter is trying to figure out how to do an internship, work in a hospital, get a publication, and take the MCAT all this summer. So I think I had to counsel with Dr. Hauser about that. So I'm here to talk about cardiac leads. I have basically spent my career at Medtronic working on material science. Soft materials is my specialty. And these are some really stark statistics, and I really have dug into these. So 40% of all of Class 1 medical device recalls, not just cardiac, are their primary cause is the material of construction. And really that number hasn't changed over the last 2 decades. And that's kind of curious to me. So I've been thinking a lot about that. And why I'm here is 2/3 of those involve polymers. It's kind of crazy, but we need these soft materials to make complex medical devices. And my hypothesis is that we face really two main challenges in predicting those properties. One, we're a victim of our own success. These therapies have gotten so much better. We're now being asked to predict the durability of these materials over time frames that exceed a decade. And if you think about in your life when you've used a highly complicated electrical device, how many of you have even owned a cell phone for more than a decade or a TV? I mean, these are really hard things to do. You know, only my children own cars that have lasted more than a decade, right? And so accurately predicting that performance over decade plus lifetime usage is really a scientific challenge. So it's not that we've gotten worse, it's our therapies have gotten better. That's my thesis. But let's go back and look at some of the things that we could do better. So I've just taken a few medical device, those Class 1 recalls, and summarized them there. As Dr. Hauser mentioned, we have been struggling to improve the durability of our insulation materials for decades. And so one of the most consequential recalls for Medtronic in proportion to its size at the time was MIO, metal-induced oxidation. And essentially, we did all this work in the laboratory and under-predicted the rate of degradation of that outer insulation. We can move to more modern times, metal-on-metal hip implants, new alloy, really studied the loading conditions and the abrasion, but again, under-predicted the results that actually occurred by more than 50 times. And finally, we're gonna spend some time talking about hydrolysis of an outside insulation. Again, we under-predicted it. And why did we under-predict? It's because we have concurrent chemical assaults, and oftentimes we only focus on the main reaction and don't think about both of them together. So in MIO, we really focused on the polymer, but it was corrosion of the metal component underneath that contributed to accelerating it. So metal on metal hip implants, we focused on abrasion. This is a Johnson & Johnson, so we doesn't mean Medtronic, but there was corrosion of that metal that over time you can do all of those cycles to failure. If you do it fast enough in the laboratory, that corrosion doesn't have time to play a role in that combined effect. And again, hydrolysis of silicone, urethane materials, we were really, really trying to guard against this oxidation and this failure mode, and we forgot that both oxidation and hydrolysis play a role in vivo. And so in all of these cases, we have to then couple the mechanical, so we just talked about multiple chemical assaults, and now we need to couple the mechanical. So these things are really complex devices. We do a really good job at accelerating mechanical, right? So I can do 15 years of testing in a half a year, but how do I get to understand that mechanical as a function of that chemical assault? And that's where we really focused our research. And Dr. Hauser said it in the introduction, that outer insulation is so, so important to that lead reliability. And you guys will all giggle in this classroom. When I give this to non-CART people, I always make the analogy of your iPhone charging cable. You know, you need that outer insulation. And it kind of sort of works, but it's not gonna work for long. And so that is the tricky thing about this. And predicting that behavior. So we have a long history of understanding the failure modes and building leads to have outer insulation that remains of high integrity. But I want to tell you some more recent research that we've done. So silicone, we were making leads with silicone. These are bulky, they're prone to abrasion. We already talked about metal ion oxidation, or MIO. Really, we've come together to build these very complex composite structures where you see inside this Sprint Quattro lead, you have the silicone inner, we have some fluoropolymers that wrap our conductors to prevent those ions from reaching the polymer, and then we have that polymer outside. But we have gone back and we've gone back into our treasure trove of return cardiac leads. And I'm just showing you some pictures to understand that environmental stress cracking, or that whitening that you often see on a lead when you explant it from a human. And what we discovered are two things. First, when we took a collection of leads that have been exposed for 5 years in the body, 10 years in the body, and 20 years in the body, we could find parts of the lead that looked pristine. And other parts had this opaque white look. And we were really curious about that. And we went deeper. And this transition between white and pristine can happen very, very abruptly. And you find places like that picture in the middle where it's very heterogeneous. And what we were able to do, if you look at this white, this white cross-section here, we were able to cross-section that lead. We embedded it in some epoxy, and then we were able to go in and look at the tissue contacting surface of that polyurethane as opposed to the inner surface. And then we were able to go with an experimental technique called Raman spectroscopy, and we can distinguish between oxidized polyurethane and unoxidized polyurethane. And what you see is I've colored the oxidized polyurethane green and the unoxidized polyurethane blue. And we noticed that we had these white areas with all of this surface topology, and they're white, they refract light, they're not a smooth surface, but that oxidation layer is really quite, it's like the perimeter of Lake Minnetonka, right? It moves around that surface, but it doesn't seem to get thicker with time, which we didn't know that. We didn't understand that. And then these pristine areas, that oxidation is still happening, except we're not seeing that tissue go away. Right before COVID, we were working with a doc out of St. Paul, and he harvested 2 human leads for us where we were able to gather the tissue over the surface. And I'm showing The tissue that was found over a pristine surface, pretty acellular. This is not science. This is an N of 1. This is not even a correlation yet. We're hoping to pick this work up. But this area over this lead that looks like those macrophages have come in and eaten it away almost. It was very active. And these were both 15-year leads, so 15 years in the body. So we're just beginning to learn about oxidation. But when we think about these current reactions, oxidation is very superficial. So I have a lot of really good material underneath that oxidized surface that can bear the load of that mechanical displacement. So again, my hypothesis, early diagnosis and our ability to make better leads. So up until the 1990s, as you know, we weren't extracting a lot of leads. Those leads didn't need to come out because our patients really didn't survive that long. And once lead extraction began, now we needed to really think about making leads for longer and longer durability. So my team has an aspirational goal of creating a 50-year lead. But when you just look from the 1990s when we first started extraction, these dots I placed on here are based upon when we design a cardiac lead, what do we think end of life looks like, right? So right now, we're designing our next generation high voltage lead. It's intended to be a 20-year lead, right? That's pretty incredible. How do we know it's gonna last 20 years is where we're gonna go. But when you look at longevity advancements, along with miniaturization, we're making huge strides in how long our materials are lasting in these leads. But we are still seeking, if I wanna get to that 50-year aspirational goal, we think we need new materials. And how do I get a new material and test it and know it's gonna last for 50 years? This is a trick, right? And we do not wanna put it in people and wait, right? We wanna test it in the laboratory and bring it in. So one of the things that we often look at is when we take a new material and put it on a device, we have certain guidance documents we sell all over the world, from the FDA when we sell in the United States, to the ISO standards that are more worldwide, to the very brand new European Union medical device regulations. So when we take a lead and you read these documents, everyone says you gotta put the lead in an animal model. The hypothesis is that the in vivo environment is so darn complex that we can't simulate it in the lab, right? But let me show you a 5-year canine implant. This was a new material. We put it in dogs for 5 years. Now, 5 years is a long time to wait to see if a new material is gonna be adequate, right? Most industries do accelerated testing. When you buy your car, they didn't wait 5 years with your car in salt solution before they knew if they were gonna sell it to you. Now, your car, you can go and you can get a panel replaced quite easily. These are hard devices because they get put inside you and we don't ever wanna replace them until their end of life. But when I take this animal and I look at the extent of my chemical reaction, if I'm now focused on these chemical reactions, and I look at the time it was in the animal, 5 years is a long time. These are the numbers I have in terms of measuring the reaction on that new material. And I had a statistician draw a blue box around the 95% confidence interval. So a bigger slope means more reaction. A flatter line means less reaction. And that's gonna be, I'm gonna show you several of these plots. Now let's look at our guidance. So really, I put this in for 2 years, and either my material's really, really good or not so good. I put it in for 5 years. It doesn't really teach me that much more. It's either gonna be really good or it's not. And that's not a guess that we wanna make, right? And if I look at these guidances, the FDA guidance written in 2000, ironically, that gives you 2 choices. You can either put you can either do an oxidation test and rule out MIO and ESC. And if it looks good against oxidation, you only need to put it in for 6 months in an animal. And if you don't see anything, you're good, okay? But that's a 30 times extrapolation for a 15-year lead. And I just told you we're doing our next generation lead to be a 20-year lead. When you extrapolate high variable, low number of replicate data 30 times, that, I mean, a statistician would tell you that that would be a big no-no. So let's move to the more recent ISO standard written in 2010. This standard says, you know what? You can do you can actually expose your material in vivo or in the laboratory. It's 70 degrees for 60 days, 2 months. And if that data is confusing to you, then you can revert back to a 1-year animal data set. Okay? So then that's a 15 times extrapolation. Again, animal data inherently, they're expensive. We can't go look at it every day. We have to take down that animal and look at it. So we have high variability and low statistics anytime we go to animal. And the EU MDR, they basically, this was written most recently, they basically say, oh my goodness, this is really hard to do. Figure it out. You need to figure out what end of life is, but we're not going to tell you how to do it. Figure it out. Okay, so this has been recognized in the standards as something that's very difficult. So what we're trying to do is bring methods to that. So I'm going to simplify the world here a little bit, so let's go with me. When you put a material in the body, there's 2 main reactions, hydrolysis and oxidation, right? I showed you some of that oxidation. You get these rough surfaces, you get opacity. A lot of people, to measure oxidation, have just looked at surface roughness with optical microscopy or scanning electron microscopy. But we now know that that doesn't tell you about oxidation because there's some parts of that fibrotic capsule that maybe don't give you that surface roughness. But that's where the industry is. You can look back at these references here. But hydrolysis is a bulk reaction. So the water permeates the bulk of that polymer first and reacts with every single bond, as opposed to macrophages that sit on the surface and spit out their debris, their superoxides, and those oxides are very, very reactive, and they react before they diffuse in. Water is not that reactive, but there's enough reaction when we're talking about 10 years, 15 years, 20 years, that it reacts in the bulk. And so we tend to see smooth surfaces, and we tend to see bulk failure because we don't have good material sitting underneath to support that mechanical load that we talked about. So when you look at these reactions and you think about how you're going to predict long times, there's really only 3 ways to do it. The first way is the material can react super fast. You can see it in that ISO standard 1-year assessment. It's 60 days at 70 degrees or 1 year in the body, but we don't want a material that reacts that fast to create a 50-year lead. So that's not going to work for us. You can the next way to do it is to have a measurement method that is so precise that you can detect a change without a lot of variation, and you have confidence in your extrapolation. Okay, well, we're not in my business into making new measurement methods. So we really only have the last option. We can accelerate the reaction in a way that we see a big enough change that we can start to extrapolate. And so this is really the strategy that we took, is we're trying to precisely accelerate that reaction to measure the changes and have confidence in an extrapolation to 50 years. So let me just show you high level how we did that. So when we take a polymer, these are long chains, so you can think of them as like a bowl of spaghetti. And when that polymer reacts, the only reaction we care about are the reactions along the backbone, because those polymers get their strength from entangling with their neighbors. So you kind of think of, I know we're in Minnesota, there might be some fishermen in the audience. this long fishing line getting entangled. It's hard to, that's how those polymers get their strength, is entangling with their neighbors. But when they start to become shorter and shorter, they lose their mechanical strength. So if you're going to predict a rate of a chemical reaction, we have to count number of reactions over time. You guys remember that from your chemistry class? So that's a rate, reactions over time. So if we measure the length of those polymer chains over time, one reaction reduces the molecular weight on average by 50%, 2 reactions on average to a third, and so on and so forth. If we plot 1 over molecular weight over time, that's a rate. So that's what I'm calling this Y ordinate. That's the extent of chemical reaction. I just saved you from the math on there. So we can measure that chemical reaction at many different temperatures. This is what you take advantage of when you put your food in the refrigerator so you don't have your food spoil. You slow that reaction by cooling it down. We're doing the opposite, speeding up that reaction by heating, okay? And so what you have to be careful about is you make sure you don't want to turn on new reactions. You want to make sure when you heat that material up, the same reactions are happening that happen at 37. And we have ways to probe that chemistry to understand that. And if you create this series of curves, you can then use the Arrhenius equation to manipulate those curves and make a reduced time plot. And what I want to point out here is you can see the black data is at 37. We could have either extrapolated the black data. That's dangerous business as we go 15x, 30x. But we can use the higher temperature data to make sure that extrapolation is for sure linear, okay? So that's what we've done. So we did that, and we have been working for the last 5 years with the FDA to make this accelerated method a standard in the industry for introducing new materials. And you can go to this new medical device development tool program. It's been online since 2017. They've only qualified or approved 17 new standards for this. So it's pretty rigorous, but we've been able to do that, and we're happy to report, like about a year ago, we got our accelerated testing. But just because you have a standard doesn't mean the industry believes that this works. So let me go through some of the data that I did in getting this medical device tool qualified. So let's go back to polyurethane and that oxidation, whether it be environmental stress cracking or MIO. A tremendous amount of work was done in the industry to understand where those reactions were happening within that polymer backbone. And so polyurethanes are made up of soft segments and hard segments. I drew a cartoon to save you from the chemistry. The soft segments are the red squiggles on the line. And so if you remember from your general chemistry class, you have a reaction A plus B goes to C. One way to slow that reaction is to suppress the number of reactants, right? So if I have A plus B goes to C, and I don't give it enough B, that reaction isn't going to make as many C's. So if C is my short polymer, I now want to take away red segments, right, to turn off my oxidation reaction. And what the industry did was to substitute a new red segment. So they took silicone and substituted in silicone. And then the materials manufacturers peddled that to all of us device manufacturers, because we don't make our own materials. We buy our materials, and said, look, we have eliminated a good portion of the oxidation reaction. It will slow the reaction. And so this is a great marketing tool that they put in to market to us. And if you take silicone and all the things you love about silicone, and you take all the things you love about polyurethane, you're going to get this magically new material. So Dr. Hauser mentioned my advisor, Frank Bates. Frank Bates is a copolymer guy. That's what he's known for worldwide. And he's like, wouldn't that have been great? You would have graduated with your thesis so much faster if you could have only gotten all the good properties when you combine two things. But it never works that way. So if we go back to this guidance, Boston's being peddled this material, Medtronic's being peddled this material, St. Jude at the time is being peddled this material. And if you go back to the guidance, what the guidance says, well, we don't want MIO or ESC, so you got to do this oxidation challenge test. And if it looks good, if you don't see all these cracks, if it looks better than what your old material looked like, then you only need 6 months in vivo. It literally is the law, it's the rule. And they did it. And it looked good. We did it. Except we decided 6 months wasn't enough for us for a new material. We were a little nervous about that. So we put it in the dogs for 5 years. And when we opened up those dogs, this is what we saw. We're like, oh my goodness. This is not what we want for lead insulation. So this looks way worse than my iPhone charging cable that's kind of sort of works, right? It kind of sort of works. So not only were we working on this deeply, our physician community was working on this. And this is a paper by Dr. Hauser, because while Medtronic took a huge pause at that point, not everybody did. Okay, so these went into patients, and docs were coming out saying, this doesn't look quite right, right? But here's what was going on. This is 10 years of clinical observation, and we're behind the scenes doing our work saying, I really need a 50-year lead. I need a new material. This seemed like a good idea on the surface. Like, what can I do to learn from this and design a new material? Well, I have to be able to predict this in the lab, because I'm not going to put this in the clinic to wait and see. First of all, I'm not going to be here in 50 years, like working my day job, right? I hope I'm here in 50 years, but not working my day job. So I want to do this before I retire from Medtronic, right? So we published this accelerated prediction back in 2012. And so we took that silicone urethane, I'm going to call it a silicone urethane, where we've substituted out of some of those red segments for blue segments, and we've made this prediction. It goes into the peer-reviewed literature, and oh my goodness. In vitro doesn't simulate in vivo. How can you predict this long? This is not true. You turned on reactions that weren't right. So you know what we did? We kept the experiment going, and we did real time. This is the longest experiment I've ever run in my life. It's a 9-year experiment. Okay, so I wrote this. This was my COVID project. I wrote this paper. So we had, I would have liked to get to 10 years, but I was home, with COVID, and it was at 9 years. And I said, that's good enough, right? So the blue is the real time. And I had to say, I was sitting there in my home office, and I kind of did a happy dance. Like, yeah. Why were we, I knew we were good. I just didn't know we were that good, yeah. Right? But nonetheless, there were people out there that said, you know what? You basically accelerated hydrolysis. In the body, other things happen, which is like a faulty argument, because it would have only gotten worse. But we said, okay, we'll do an animal test. Now, this is data that hasn't been seen before. So we go 5 years. This was concurrent to my in vitro 9-year test. I go 5 years in sheep. Look at my data. I had 2 time points, 2.5 year and 5 year. I even put it in the brain, the heart, and the sub-Q, because I was like, it's hydrolysis. Right? Like the brain's kind of fatty. Maybe there's not enough water in the brain and it matters. Turns out it doesn't matter. Nobody's seen this data before. But then I have my low number of replicates and my high variability problem. What happens if I overlay my prediction on there? So you really need, what the in vitro brings you is not only speed. Look, I did all of this in 1 year of laboratory work, but it brings you many, many time points that you can't get in the in vivo. Right? So it offers additional information that you can't get. So the argument that this has taught, what this has taught me is, I think we need to use animals as a way to confirm our experimental results, make sure we haven't turned on any additional reactions, anything that is not going to happen at 37 degrees in vivo, but it really isn't sufficient to project long times, okay? So I believe this, the world doesn't believe this yet, right? Dr. Al-Shami, an explant physician out of Emory University, some of you may know him, he had some of those same publications like Dr. Hauser had, questioning the durability of the leads that had these silicone urethanes as their primary insulation. He came and he explanted 100 leads. He was material agnostic, and he separated those leads based upon their material of construction for their primary insulation, and he did that same measurement, extent of chemical reaction. And there's basically three primary polymers that we make leads out of. Polyurethane, soft polyurethane, ADA, harder polyurethane, 55D, and the silicone urethane, which has been trade named Optum. And so this is his human data. So these came out of humans. He sent them to Medtronic so we could identify the material of construction for him, and we separated them into the buckets, and then he sent them out to an external lab to measure that molecular weight or how much backbone chain scission that they had seen in those materials, and he plotted it as a function of human implanted time in years. Look at the number of years. So this is even better than my 9-year experiment, right? Or my 5-year in sheep. So now the million-dollar question is, how did this compare to the accelerated prediction, which, by the way, took 1 year in the laboratory, right? So there were only 2 materials that we had done the accelerated prediction on. The ADA, the softest polyurethane, that had the most soft segments in it, so it had the most susceptibility to oxidation, and the silicone urethane. So our publication on ADA was in 2014. So Dr. Al-Shami's paper came out this year, so we're like a decade earlier. This is what we've predicted for ADA. So our accelerated prediction and then our human explants that Dr. Al-Shami collected out to 16 years. And you say, you know, it's okay. But with humans, we still have that problem with low numbers and replicates. Dr. Al-Shami had collected more leads than he had a budget to analyze. So I said, send me those leads. I'll analyze them in my lab. So I added additional data points to his published data. So it's kind of like flipping a coin, right? If you flip it enough times, you should get 50% tails and 50% heads. But if you don't have enough data, it might look a little different. So I think that's what's going on here, and I think we'll converge on the high fidelity in vitro data. That's my hypothesis. But now let's look at that Optum data. So that's that prediction for Optum. We still don't have a lot of statistics. I do think if we could get more data, it would start to merge. Each one, I should have mentioned this, each one of these bars of color around the data is the 95% prediction envelope, right? So it's a statistical analysis to say this is the variation in the data and this is where you would expect future data to lie within this band. So in short, I feel like we have a laboratory accelerated prediction that we can use to look at new materials, which is really exciting for me. And we can start to not only look at new materials, we can start to really understand with these accelerated conditions, if I analyze a material way out here, my signal to noise for what that chemical reaction is going to be much, much higher. So I can now go in and tinker with those molecules and make better linkages to connect those soft segments and hard segments. So the question is, we need to understand what chemical linkages are breaking. And so we've been using this accelerated technique to go in and look at different materials. And what I'm showing you here is that polyurethane material. And it's the exact same chemistry. We've just substituted out some segments for that PDMS or those blue segments. Is this a universal thing? Right? Is this going to happen if I put PDMS in a different polymer? Because it's actually kind of an interesting idea. And so we went to polycarbonate urethane. and then a PDMS modified polycarbonate urethane. And so we're kind of curious about this. I'm giving you a little insight to my current research. I'm kind of curious about this. Like whenever I put in PDMS, I double, or, I'm going 7 times faster in the polycarbonate. And so we have a hypothesis that we're testing, and we think if we come to some clarity on what's causing this acceleration, now we know what the problem is, and we can design new molecules to eliminate that reaction pathway. And so what we think is we're creating these bonds. These two materials don't really like each other, and there's a lot of molecular stress there, and you put in that water, and that stress bond is the one that's breaking first. And so we have evidence that we're breaking at the interface between the PDMS and the other polymer. But nonetheless, I think that this is the way to move forward to create new materials. What this is also doing is, remember that EU MDR, that there's surveillance requirements, 11 references to surveillance, but we don't really know how to test that. So as we've brought in modeling and simulation online, we're starting to play with looking at material properties in different designs. So I'm just taking my animal explant that I showed you earlier. We had put in 2 different designs into the animals. We had put in this top one and this bottom one. And if we understand that splitting behavior we observed, we hypothesized it was kind of like when you bend your finger, you create wrinkles. The peaks of the wrinkles are high stress areas. And when that material weakened with reaction, we hypothesized that was causing the flaw that was then propagating as those splits along the lead. So you can model this, and you can put in different cross-sectional designs, and we can calculate the design margin, or the safety factor. So this is an engineering term. When your safety factor is above 1, you shouldn't fail, ever. And when your safety factor is below 1, you should fail. This is engineering principles. And so each one of these safety factors was calculated with time equals 0 properties, okay? And when we put these in these animals for 5 years, this was our planned design. We never put that into our animals. We put the top one in and the bottom one. Only the top design failed in those 5-year animals. The bottom one didn't, but this had a much bigger design margin. So what happens when I put my time equals 5-year properties, which I now know because I can do that accelerated testing and then measure the mechanics, into these same models? So my planned commercial design would have failed at 5 years. I observed my top design fail within 5 years, and I did not observe my bottom design failing within 5 years. So we're starting to build this modeling and simulation where we can take the chemical reaction and the mechanical response based upon that chemical reaction. And this allows us to go through design iterations in a much, much faster pace. Okay. So I think that this accelerated testing, in summary, gives us great ability to both design new materials and design devices that are resistant to whatever material liabilities that we might have, right? They might not be the smallest diameter or whatever, but at least we will go in knowing what happens in vivo. And I really do think that the qualification of this MDDT by the FDA is setting forth a precedent that accelerated testing, predictive testing with today's longevities actually needs to be part of the testing scheme. We're just starting to talk to the European notified bodies and the rest of the regulatory bodies around the world. It takes time, but FDA often leads the world with this. So to reiterate, when we put devices in people, this is another Dr. Al-Shami publication where he's looking at electrical performance of more than 8,000 cardiac leads. And prior to this MDDT, we had the required preclinical testing of all of these materials in that highlighted area. So it was really hard to distinguish if it was better or worse than what you already had. even if we go out to my 5-year extended, which is not a rule, right? We just did it. It's not a rule. You could argue, given the variability that I showed earlier, that it's really not that different than your predicate. And the accelerated testing allows you to take things out much, much further in time with enough precision that you can discern differences if they're there. So I think that this changes the thought process. I think the term biostability gives us a false sense of security that we can have a material that's biostable. We test it at time equals 0, and we assume that material doesn't change its properties. To create a 50-year lead, that material will change its properties. It absolutely will. So we have to understand how and make the design accommodate that change. And then finally, the accelerated testing scheme, as I just showed, allows us to simulate performance at a future time in the device, which is really exactly what we want to do. Who cares about the material? It's how that material interacts with the design and how that performs for the patient that we care about. So with that, those are my prepared remarks. I would be delighted to answer any questions that you have about the work.
Going back to 2012.
Yes.
I remember meeting with you in 2012 and with Jennifer Miller and actually at another point in time, Frank Bates. And you at that time had identified the critical reaction where the material was failing, the optimum, I'm just going to call it optimum material. And what you described was hydrolysis of the chain. And there was a very interesting picture of spaghetti where if you have long spaghetti, you can pick it up and nothing falls apart. If you have short pieces of spaghetti, you can't pick it up. But it was back then that it was, to me, it was pretty obvious that that material was not something you wanted in a pacing lead.
Absolutely.
So, but it took you 10 years to demonstrate that convincingly with accelerated testing and finally with the molecular weight of the implanted leads.
So it was a long time. But I do want to correct one thing. Your story is 100% true. But you know what? What I didn't tell you is we had a program in flight. It was a 7 French high voltage lead. We were going to call it Savento. And we saw this animal data 6 months before our clinical launch. Mm-hmm. We were gearing up to make this lead. And so it didn't take me 10 years to convince my internal folks that this was right. We did not launch Savento, and as you know, we're still using a 25-year-old high voltage lead because the performance is so darn good. It's hard to come out with something that is inferior. So I think the tricky thing and the interesting thing about this exercise is oftentimes when you decide not to do something, you never know if it was a good decision. Here, like I said, we can kind of say it was a good decision not to do it, but had somebody not put it in the clinic, we probably wouldn't have done all this work to close the gap and get this guidance changed. We might have waited for that next material failure. I don't know.
But that observation did prevent Medtronic. And by the way, when Medtronic market releases a lead,
we're big.
It's taken up by tens of thousands within weeks. But that one observation prevented Medtronic from making a commitment to a lead that.
It was not easy internally. I mean, I was a little stressed out for a long time.
But now there's the OmniSecure.
Right.
Okay.
Right.
I mean, that's another story.
Yes.
Any questions?
It does not use this material, by the way.
The room is mic'd, so.
Oh, it's Mike?
Yeah.
Oh.
Thank you for a great talk. I really appreciated that. When you are building your predictive models, especially with oxidative stress, how do you account for changes in the chemical environment, such as with an open heart surgery?
I showed you a little taste of the research we're doing with trying to get fibrotic capsules right next door, and I'm not even sure that human variability. I think the oxidative stress, I was really worried about it. Of course, it was one of our biggest recalls. And the reason MIO, that metal-induced oxidation, was such a big recall is because it caused the reaction to be uniform throughout the thickness of the material. ESC is so superficial. As long as you have decent thicknesses, I mean, I take a little ESC with bulk polymer underneath all day long versus a through reaction. So there's a doc that just wrote one of our external research proposals who wants to study exactly what you just said. Like, how does oxidative stress change based upon the patient's drug regimen and their fibrotic capsule? And I think it would be fascinating, but it's only the first 5 microns that oxidizes, and it seems to be very, like, it happens all within the first 5 years and then it stops. And I'm not sure that that's the limiting factor of preventing my 50-year lead. Like, I just don't know, but there's so much to learn on that question. I don't know the answer.
You have somebody online?
We do, yes. Thank you very much for that excellent presentation. We have one virtual question from Dr. Josh Buckler. His question is about AI, and the question is, are you starting to utilize AI to help in predictive modeling and for new material discovery?
Yes, that's a very interesting question. That, I think if we fast forward 10 years, we'll be able to do that. I think we're still doing the science to understand the technical inputs to run the AI algorithms. AI works with big data, and we don't have big data yet to feed that AI mechanism. But I do think that fast forward, we'll be able to do that.
Thank you.
Other questions? Thank you very much.
Of
It's been
course,
wonderful.
thank you for inviting me.